All right, everyone, we're going to go ahead and get started. Thank you so much for being here for our seminar. My name is Cinnamon and I'm one of your hosts, but most of this work and today's speaker are hosted by our student group, HSO. So they will be a part of this conversation as well in just a second. But I wanted to welcome everybody here and say thank you for coming in from the sunshine to spend a little time with us. For folks that maybe haven't been at one of these seminars before, these are hybrid events. So we have folks online and folks in the room. For folks online, please put any questions you might have into the chat box, and our volunteer Roseanne will help navigate those or be able to talk to our speaker and make sure that your questions are answered. For folks in the room, if you raise your hand, if you have a question, I'll bring you this mic, and that way folks online can hear as well. But again, thank you so much for being here. Quick announcement, I'm excited to talk about next week's seminar as well. So next week's seminar is on May 8th. In this room at the same time, but May 8th, we have folks that have been working with Hilary Baudet from the OSU School of Marine Policy and from the University of Delaware's School of Marine Policy, Marine Science and Marine Policy, and they're going to be here to talk about community benefits and impacts from offshore wind development. So that's been one of those topics that's been spinning around here for a little while, so I'm really excited to have them come and present. But for today, like I talked about, the speaker today is our student invited speaker. Every year, our students get together and vote on a speaker that they think would bring an important voice and perspective, not only to their work, but also to the broader HMSC community. These speakers do a whirlwind visit with us, talking to student groups, meeting with both researchers and students one-on-one, and giving a seminar like the one for today. So I'm going to hand it off and let HSO introduce.
Hi, everyone. I'm Susie Winquist. I'm a PhD student in the Seabird Oceanography Lab, and I'm representing graduate students here at Hatfield today to welcome our student-invited speaker, Dr. Kyle Elliot. And yeah, we just really want to say thank you. I know it's a long trip from Montreal to come out here for a whirlwind visit, but yeah, we really appreciate having you here, and we're really grateful and excited. So thank you. We also want to thank the director's office for supporting this event. They support us to bring a speaker here every year, and we're particularly grateful this year. They allowed us to bring an international speaker, and I know Canadian imports are a little expensive these days, so thank you for making this one possible. And yeah, with that, I guess I'm just going to introduce our speaker today. So Dr. Kyle Elliot leads the Arctic Ecology Lab at McGill University, and he currently holds Canada's Research Chair in Arctic Ecology, is a board member of the Society of Canadian Ornithologists, and serves as on the editorial board of the Marine Ecology Progress Series. His path to biology began with an undergraduate degree in mathematical physics and conservation biology at the University of British Columbia, and he went on to earn a master's degree and PhD from the University of Manitoba. After his PhD, he held a Natural Sciences and Engineering Research Council postdoctoral fellowship at the University of Guelph, and after that accepted his current position as a professor at McGill University in 2015. Quick note, before I hand it over to Dr. Elliot to talk about his really exciting research on seabirds in the Arctic. After the seminar, HSO is hosting a happy hour at Pier 1. So we'll be heading over there right after seminar around 5:30. So we hope to see many of you there. We have a little certificate of our appreciation from the student organization to present you with. All right. So this is, just to say thank you so much for
Thank you. That's really nice.
Yeah, and with that, I'm going to turn it over. So please join me in giving a warm welcome to Dr. Kyle Elliot.
Okay, well, thank you, Susie, and thank you to the students for hosting me, and thank you to Katie as well. It's a real pleasure to be here, and especially on a day like today when I hear the weather's always like this, and but it's certainly nice to be here from Montreal, one of those rare Canadian imports. Yeah, today I'm going to talk about seabirds as indicators of environmental change. And I think this kind of image encapsulates what I'm going to talk about today. You have 6 thick-billed murres flying back from the flow edge. And some of them are successful. You can see they're holding fish in their mouths and their beaks, and some of them aren't. And so we can ask ourselves, what that might indicate. Perhaps it tells us something about, the distance to the flow edge, how the ice is distributed. Perhaps it tells us something about the prey themselves, the abundance of the cod, or maybe it tells us something about, some other parameter like, the contaminants or the levels of compounds in the fish themselves. And so I hope to, explore today some of the ways in which we've been trying to look at these types of questions. I'm going to pretend like this is my work today, but actually this is the work of many people that I'm fortunate to work with. We call ourselves the lab with loggers, and if you look closely, every member of the lab has a little heart rate logger on their in their front pocket. Yeah, we do lots of logging, maybe not that type. I also want to acknowledge that on the work that I'm going to present today occurs on land owned by Inuit or in Alaska by the Chugach First Nation. And although I'm going to ignore it in the talk today, I'm going to provide kind of a Western science perspective. There are, of course, a long history of deep knowledge about the systems here, about the seabirds there, and including we've been working with Josiah for 30 years now. And he just has a tremendous amount of knowledge about the system, the broader system, and the social ecological environment that we're working within. Okay, so the question I'm going to try and tackle today is, how can we measure environmental change, right? And one way we can do that is using kind of an umbrella species, or I'll use for most of the talk this kind of idea of an indicator species, but that's pretty much the same idea as an umbrella species in many cases. I mean, maybe what we're less interested in is whether or not the seabird is directly telling us how much plastic is in the ocean, as it in fact just kind of being something that we care about, and therefore something that we can use to as an umbrella for other species. And so an umbrella species should be telling us about ecosystem function. They should be species which are easily monitored, that integrate signals over time and space, or typically top predators, or species which are covering large areas of space and perhaps living long time periods, and that are of course sensitive to change. And there's lots of good examples here of that. And I'm going to hopefully convince you today that we could add seabirds to that list. So brings us to the kind of idea of an indicator species, which is a type of umbrella species perhaps, or at least related to that signals a change in the abiotic or biotic conditions of an ecosystem. So it's telling us something about the changes in the ecosystem. And perhaps the best example of that, at least in the seabird world, would be chemical pollution. So, seabirds as indicators were there right at the start of ecotoxicology work on, brown pelicans, for instance, 50 or 60 years ago, now was telling us about the state of the marine environment and about levels of chemical pollutants there that then led to, policy changes. And of course, this is very important because piscivorous predators are often good sentinels for human exposure. And so they're telling us about what, about the levels which we might be exposed to. they're also really good indicators of the physical environment. I just wanted to highlight some of the work here happening at the marine station here. Using them as indicators of physical changes as well. And so, some great work happening, looking at seabirds as indicators of physical change. I'm sure you're familiar with. So. We have this idea then that top predators like seabirds integrate information over time and spatial scales. And this is some work we did a little north of here off of the BC coast. And you have 3 different species here. You've got the cormorant, which is covering the nearshore environment. You have the rhinoceros auklet, which is a little bit further offshore. And then you have the Leach's storm petrel, which is feeding well off the continental shelf. And so you've got 3 species which are telling you a little bit of different story about what's happening in terms of the indicating something different about what's happening in the ecosystem in terms of chemical pollution. So we then went out, collected the eggs from the burrow, and you can see classic signals like this one here for DDE. The level in 1970, and then the dramatic drop and decline in DDE over the next several decades, and of course that's indicating something about the environment and about policy change. Something that we did and how it's affected the environment. Here's the same thing for PBDEs, so a brominated flame retardant. A lot of concern of that in the 90s and into the 2000s, increasing rapidly in the environment. But then, as you can see, tailing off in recent years. And you can see the exact same thing for things like PFAS, for instance, where levels have increased. We then restricted them, and now we can see in the eggs that they're decreasing. So a nice example of how we can use seabirds to indicate change, both in response to our policy, but also kind of then what happens in the ecosystem. And you see it first in the cormorants, then you see it in the auklets, and then you see it in the storm petrels. So they can tell us something about chemical pollution over a time scale, but also over spatial scales as well. And so I think this is kind of one of the exciting areas that ecotoxicology is going to. Recently, it's work that we did with the group in France, the ArcTox group, led by Céline Albert. And each of these points are one of 7 species which were tracked from, I think it's almost 30 colonies across the North Atlantic, including some colonies in Canada. And you can see where they went in winter. Because, of course, one of the big critiques of using seabirds as an indicator for chemical pollution is that they're migratory. And so that the signal might not be representing a local signal, but where they went in winter. But here we're able to kind of tackle that directly because we know where they went year-round by attaching little geolocators to the legs, and able to see where they might be getting this signal. So we attached the geolocator, they came back, we took a feather, and depending on which feather we took, it could either give us a breeding season signal if it was a wing feather that was molted just once a year, or it could be a winter ground feather if it was a throat feather, which is molted in the spring. And then we measure levels of mercury in the feather, because the mercury is excreted typically into the feather. The methyl mercury attaches to the cysteine, which then attaches to the keratin in the feather. And we can put that together to get a map of the entire North Atlantic. And you can see that there are some places where mercury levels are a lot lower. So birds wintering in the Barents Sea had relatively low levels of mercury, whereas those wintering in off of the east coast of Canada, off of the Grand Banks, or further north, had much higher levels of mercury. So here we have an example of seabirds telling us spatial distribution of chemicals and where mercury might be. And this probably doesn't represent what is happening in the water. Like if you were to go out in a ship and try and measure the level of mercury in the water, you probably wouldn't see the same pattern. I know that you wouldn't see the same pattern. What it's telling you instead is the level which are in prey which they're feeding on, and so perhaps levels which are indicative of other seabirds, other large fish that are foraging in the same areas. Okay, so I mean, we spend a lot of time in my lab trying to think about these types of questions, but I really think that's a really good example of the application of seabirds as indicators. What I'm going to focus on today is what they might tell us more broadly about marine ecosystem health. So trying to kind of apply some of these ideas about that work so well for ecotoxicology and trying to apply that to broader questions of ecosystem health. So the idea then is that you can take your icebreaker, which this particular one costs $150,000 a day and gets only a single track, or you can get my icebreaker here, where they work for free, or at least for a little bit of capelin that I don't have to supply, and they spread out over a 300-kilometer foraging radius and do that continuously. So, this is the idea that we can use these seabirds to be able to indicate something about the marine ecosystem in the area around the colony, at least. And I'm going to argue that these are, the seabirds are also really good because they have really high energetic costs. So this bird here has the highest wing loading of any bird, which means that they have, it's just a fancy way of saying that they have small wings and a big body. And this means that they have really high metabolic costs. So at the very top there, you see a murre as a multiple of basal metabolic rate. That's 30 times BMR. That's incredible that they're actually able to sustain that for hours at a time. This is what Usain Bolt is able to sustain for his 100-meter dash. But humans are not able to sustain anything like this for more than a few seconds. And because of that, because they have such high energetic costs, it means that they're going to be doing things very efficiently, right? They can't afford to, stay out in the lobby and drink some coffee. They've got to do things really, really efficiently, right? So, in principle, at least, they should be really good indicators of food supply because they're working as optimally as possible. So, these ideas have been around a really long time. Maybe some of the first formulations of this were by David Cairns, almost 40 years ago. And, at that point, David was working, on colony-based measures, right? He was like, what can I get when I'm just working at a seabird colony? And he came up with this idea here that you've got, you can look at survival, and survival should be only impacted when food density is really, really low, followed by, breeding success, chick growth, colony attendance, and activity budgets. And so depending on what you're looking, what level of food density you're looking at, you can have different parameters that might have different sensitivities to food availability. But the idea is really simple, that you could use seabird breeding success to represent the amount of fish. So if they're successful, you have many fish. If they fail, there's few fish. And then 15 years ago, there was a team that kind of looked at this from a bunch of bunch of colonies around the world, and you can see that well, I mean, it's certainly true on average. It certainly works, right? As soon as you get to a food density of about 1/3 of the maximum level, then breeding success decreases. But there's a lot of noise. I mean, it's not like you can look at breeding success and then tell someone how much food there is out there or what the fish density is. Well, I want to argue that the key parameter should be when the predator meets the prey. So, that's what we really need to know. Rather than focusing on or trying to look at what is happening at the colony, we really need to go out and find out what's happening at that moment where the predator captures the prey. And so we tried to reformulate that a little bit. On the left side, the dashed lines are the kind of theoretical predictions. The dark lines are what people have actually measured. And, so there's the classic, Cairns idea on the left side. And we kind of argued in this paper here that maybe not energy expenditure, because energy expenditure could either be high because it's they're working really hard to get a lot of food, because food is not very available, or it could be the other way around, right? But if you look at foraging effort or energy intake, this should tell you something direct and perhaps linear about the abundance of food. So this is the overarching hypothesis I hope to convince you of today, that seabird energy expenditure and intake can tell us something about ecosystem change and tell us a little bit more than we're able to get from just studying seabirds at the colony. But before I do that, I've got to kind of explain a little bit about some of the techniques that we're going to use. So I'm going to explain, tell you a little bit about how we measure energetics in seabirds. And I think this is kind of a ambitious, right, to be able to go out and measure energetics in wild birds and be able to tell you something about prey. But I thought that was relatively ambitious, but not quite as ambitious as these people here, who wanted to study the bioenergetics of God. But you know what? Actually, there's a region of Japan where they venerate the cormorant as a god. So maybe I am going to tell you a little bit about the bioenergetics of God. Okay, so the way that we're going to study energetics is by putting on biologgers, particularly accelerometers. And so here we have a GPS accelerometer on the tail of a black-legged kittiwake as it's jumping out of the window at my study site. And this tells us, right, it's triaxial, so it tells us the acceleration in 3 different axes. And that allows us to figure out body position and also how much movement is happening. Of course, it's exactly what I have in my phone here, and tells me how many steps I take in a day, and as well as whether or not the screen is up or down. So it's the same type of principle. And one of the first things we can extract from this is this thing called dynamic body acceleration. And the reason that we're interested in dynamic body acceleration is because we think that energy expenditure, like if you kind of remember back to your kind of high school physics, right? Energy expenditure is equal to work, which is integral of force times distance, which is integral of mass times acceleration over distance. And so that's then mass times speed times the integral of acceleration over time. That integral of acceleration over time is what we call dynamic body acceleration. And if you didn't follow all of that, I sketched out rather poorly last night, but I tried to explain how this was. So here you have the X, the Y, and the Z axis of a bird that's flying. Then it's calm for a little while, and then flying, and then resting for a little while. And the area under that curve is your dynamic body acceleration. And so you have periods of low dBA when it's resting and high dBA when it's moving. So you have, so it's pretty intuitive, right? I mean, there's periods when they're working hard and they expend a lot of energy, and periods when they're not working very hard when they not expending a lot of energy. And so 20 years ago now, people were able to test this out and show that it does work on a treadmill. And so this was work by Rory Wilson on cormorants on a treadmill, and showed that, yeah, overall dynamic body acceleration does predict oxygen use, again, in captivity. But we're really interested in how this works in the wild. So we'd like to know whether or not dBA predicts energy expenditure in wild birds. And so you can imagine that in wild birds, temperature is changing, so there's thermoregulation to worry about. There's digestion. There's a lot of other processes that could overwhelm things compared to the really controlled lab environment of a bird just walking on a treadmill, right? And so we went out and we measured energy expenditure in 5 different species. And we use doubly labeled water to do that. So we injected the birds with doubly labeled water. We took a blood sample 48 hours later, and we compared that to the predicted energy levels from dynamic body acceleration. And there's also 3 other studies which did a similar thing. And the simple answer is yes, that DBA does work. It works really well in seabirds. And you can see across all of these different study systems, all of these different species, that there's a decent correlation between dynamic body acceleration and daily energy expenditure. And the situations where it doesn't work as well, such as the sulids at the right end there, have a relatively limited range of dynamic body acceleration. So I think if they, just in that study, they didn't move, there wasn't much variation. So of course, there's not much variation to explain. Okay, so that's how I'm going to be measuring energy expenditure. It's using the accelerometer to measure dBA, which we're then convert into daily energy expenditure. The other half of energetics then is energy in. And I think we can all agree, or at least the grad students in the room can agree, that the rate of food gain is important to ecologists. And so having that side of the equation is also really, really important. And people have come up with all types of interesting and useful ways of doing that. There's people have put on camera loggers onto birds and looked at what they're foraging on. For many years, people were using wiggles, and so that's not a dance. That's whether or not the depth profile went up and down on penguins, and it's sort of correlated with prey capture events. Some people measured esophageal temperature, and so that's the so the idea there when the penguin eats a cold prey, its esophageal temperature will go down. Some people used beak opening sensors so when the beak opens, they that's when they're consuming a prey item, and then increasingly people are using accelerometers. And here's an example with a spoonbill, and every time the spoonbill moves its bill, it gets picked up by the accelerometer. And so lots of different techniques. But many of them have a lot of challenges. It's really difficult. I mean, you can look at that penguin, that poor penguin, which has this a magnet and the Hall sensor there with a lead that goes back behind it. Really challenging. Likewise, the one with the temperature logger is also very technically challenging, as you can imagine. The camera logger typically only lasts for very short periods of time. So I really think the future of this is using accelerometers. And I put this in here for Susie, actually, because this is what happens when you ask a grad student to watch videos of birds eating prey and try and line it up with accelerometers. And so this is a video at the back of a...
Before you play that, I wanted
to
share. We didn't push the right button. Hang on just one second. I want to make sure folks online can... Too many things to click.
Yeah.
Okay, now go.
Great. Okay. So you can see the video of the bird swimming underwater, and then also the accelerometer trace on the top there, as well as the depth. I'll just let you watch it. Working really hard against buoyancy to get down. You see, every time it comes to the surface and catches something, there's a little wiggle in the accelerometer. Okay, so there's someone there that shares your pain. Go ahead. Um, and we can put that all together there. So I mean, and you can look at you know, the birds going down. You can see you know, you have to smooth out all of that variation in the accelerometer profile, and then once you've accounted for the variation, you can see that indeed the peaks in acceleration. So when they're burst, when they're attempting to capture the prey, then you do indeed detect these peaks, and that you know, is also the same time when they're catching prey. So this does work. You know, statistically, we can show that this does work. But it probably is only in capturing the attempts, not the actual amount that they're feeding on. And so we also tried this other technique from the accelerometers, and here we're looking at we think we can actually get the mass gained. And this is work by Alison Patterson, who was an OSU student here a few years ago, and now continued this with this PhD project. And she was looking at body acceleration and trying to link that to body mass. And so the idea there is that so you have you know, here it's it has to stay aloft by counteracting gravity. So that would be body mass times gravity. So, you know, mass times g. The wing beats will provide the lift. And so there has to be some way of counteracting that downward force. If body mass increases, lift must increase as well. And therefore, wing beat frequency should increase as body mass increases. And there's some physics that go into that, but we expect it to increase with wing beat frequency squared should increase with body mass. And you know, that just makes sense as well, right? I mean, if you have a heavier load, you're going to have to overcome that heavier cost somehow. And one of the easiest ways you can do that is by beating your wings faster. And so here's some actual data. Kind of put that, and you can see indeed this does work. And so we have the if you look on the left-hand side here, you have a bird that was weighed here at the colony. It then flew off with a wing beat frequency that is, you know, approximately that. And then after the trip, it has a higher wing beat frequency, so it's beating its wings much faster. And then we weigh it upon return to the colony, and indeed that change in wing beat frequency correlates well with the change in body mass, and you can see that on the right hand side as well. This is kind of done through a state space model, but you can see that the rate of mass gain through the trip, and that as at every place it stops to forage, its body mass goes up a little bit. And we can put all that together to try and map hotspots. And you can see here that there are some areas where mass gain is relatively high and areas where mass gain is relatively low. And then as we put that together year after year, we can get, you know, consistent hotspots and consistent cold spots for foraging. And why might that be interesting? Well, this is actually an area which is proposed as one of Canada's largest marine protected areas. And they're trying to figure out where within this proposed area, where to actually put the marine protected area, and as well as how to zone it. So understanding exactly those hotspots is really helpful. Okay, so I know that was a lot of background to kind of tell you what my main story is here, which is that you know, we're going to see whether or not you know, seabird energetics can indeed inform ecosystem health. And so, you know, whether or not we can use those tools to be able to tell us something that we didn't already know about these ecosystems. And I'll, you know, emphasize that we don't actually have you know, an alternative, or we have no information really on forage fish in these at these sites. And so there's no fisheries, there's very little science, you know, there's very few scientific, there's very little scientific information here for on forage fish abundance. So we're trying to apply this at a site where, you know, if seabirds are indicators, hopefully they tell us something about sites where we don't already know the answer to these questions. A lot of what I'm going to talk about today is in terms of climate change. I think this is, one reason why we should expect energetics to be a really important part of how these birds respond to climate change. And that's because energetics should become unfavorable for marine endotherms in a warmer world. You can see here the phylogenetic diversity, but basically the abundance of endotherms. So that would be mammals, but also true for seabirds. I mean, basically auks, like pursuit diving seabirds, auks and penguins occur at the poles. And one reason that they occur there is because of this metabolic asymmetry, whereas sharks kind of hug the tropics. There are, of course, many exceptions to this, but in general, it's true that, and this is probably because marine endotherms, they have a metabolic rate that's constant regardless of the temperature, whereas fish, their metabolic rate and therefore their locomotory rate increases with water temperature. And so the idea here is that as things get warmer, fish get faster, and so both the prey, the competitors, and the predators are getting faster. I'm working at two sites where I've been fortunate to kind of inherit both of these sites, or inherit projects which have been going on for almost as long as I've been alive. Not in this case, but in the next case, but for a long time, right? I mean, I was 15 when this study started. And this is the Middleton Island Black Leg Kittiwake study, which occurs on this radar tower. There's windows right the way around this refitted tower, and so with one-way glass, and so you can be working with kittiwakes that are just a few inches away from you, and they're unaware that you're there. And this was led for many years by Scott and Martha Hatch. And then I also work at Coates Island on thick-billed murres, which was started by Tony Gaston in 1981. So I've been fortunate to work with these two long-term sites where people who had more prescience than I did, or you started these long-term studies a long time ago. And in Alaska, the system seems to be dominated by the Pacific Decadal Oscillation, the PDO. And so when it's cold, the kittiwakes do well, the researchers do poorly. When it's warm, the kittiwakes do poorly, the researchers do much better. Whereas in Hudson Bay, it's really all about ice. And so these are, if you like, ice obligate species. Throughout its range, it occurs where there is ice. And so the species does well when ice is around. So my first case study then will be about these Arctic murres. And, we've all heard an awful lot about, summer sea ice extent. And you can see the difference over 40 years between the sea ice extent. And that's also true in Hudson Bay. Summer ice breakup is now 1.5 months earlier. Whereas in the early years, we used to arrive on, like, just to think of it in terms of a really concrete example, where the twin otter used to arrive on skis on the ice. That never happens anymore. We arrive on wheels because there's no ice left anymore. So the question here is, what impact does this earlier breakup have on the ecosystem? Well, one clear impact, and this would be a diet change. And this is the diet over the last, from 1981 to 2019, I had, but in fact, 2024 actually had the lowest amount of Arctic cod on record. So it continues. And so in blue, we have Arctic cod. This is this ice-associated fish. The way we think about it is typically that it's associated with that under ice ecosystem. So it's living on the copepods, which are feeding on the algae living on the underside of ice. And so a really important part of Arctic ecosystems. I mean, it's what makes the Arctic ecosystem Arctic. This key forage fish, polar bears are pretty much just repackaged Arctic cod. They feed on ring seals, which feed on Arctic cod. And same with these murres. But you can see that they're decreasing. So it seems like Hudson Bay is becoming less and less of an Arctic ecosystem and more of a North Atlantic ecosystem in the summer. Because at the same time, it's being replaced by capelin. So whereas we used to have upwards of 60% Arctic cod, we now have upwards of 60% capelin, and capelin is a typical forage fish off of Newfoundland. Oh, and I'll just show you that the Arctic cod tend to be a lot larger than the capelin as well. So you'd think that they'd prefer feeding on Arctic cod. But when we look at the correlation between the proportion of ice in July and the chick fledging mass, you can see that, yeah, the more ice, the better the birds do, but the correlation is relatively weak, right? R-squared of 0.09 there. So, climate science is inherently correlative. We can't do experiments. We can't, change the temperature of the globe or purposefully for our experiments, right? But we can certainly add strength by perhaps connecting various causal links. And so what does predict things fairly well is how hard the birds work. And so here you can see daily energy expenditure on the x-axis from the accelerometers, and you can see that predicts 14-day chick mass fairly well as adult body mass. And so there is a link between how hard they're working and how well they do. If they working very hard, that's not a good thing. The chicks don't grow as well, and the adults weigh less. There's not enough food in the system. So we tried to put all this together then into this structural equation modeling, or this path analysis, and this seemed to work much better. So although there was only a weak link between ice cover and fitness, and it doesn't matter which fitness proxy we use. This is actually a principal component analysis because they tend to be correlated with one another. When chicks aren't growing very well, the adults weigh less, survival is low, reproductive success is low, so they're all correlated with one another. So we did a principal component analysis of multiple factors, and you can see here, even though there's not a very clear link between ice cover and fitness, there is a good link between ice cover and diet, and then between diet and net energy intake, and then between net energy intake and fitness. And that net energy intake, of course, is coming from the accelerometer. So that's the energy intake based on the change in wing beat frequency subtracted by the energy expenditure from the dynamic body acceleration. And yeah, so I think here you can see a nice bottom-up pathway of links, even though you don't see that correlation from directly between ice cover and fitness. Clearly, when is less ice cover, that influences the diet, and so there's less Arctic cod when there's less ice. When there's less Arctic cod, the net energy intake is lower, and when the net energy intake is lower, then the fitness is reduced. So kind of a nice way, I think, of being able to connect it all together. Okay, so that was a little bit about the time trend. So I started this off with the idea of that seabirds could tell us something about time trends, but also spatial trends. And so that was from the whole 45-year data set. Now, if we look at spatial variation, we can ask ourselves whether or not these energy landscapes could be could be helpful. And here's an example with Andean condors, and here you can see you can look at the energy landscape and see areas of high vertical velocity versus low vertical velocity for these condors, and so areas where they do well and areas where they do poorly. And you can see that the condors hug very closely the area of high vertical velocity, which makes sense, right? Okay. These seabirds are central place foragers, and so their energetics is going to be determined by the fact that they're having to go out and forage. If they have to go long distances, they're going to have to expend a lot of energy. If they can forage nearby, they're not going to expend much energy. And And so here is our relationship. I'm showing you energy intake here. So this is energy content of the prey on the x-axis, and energy output, which is the amount of energy it takes them per day to get that particular energy, bring that energy back to their offspring. And the idea here is that they have a trade-off to decide on. They can either go far and get a lot of energy, or they can stay close to the colony, but then they get less. And one way to think about that is that they can forage near home and go out and just go around the corner and get a megamphipod, but there's not a lot of energy in that. Or they can go far away and kind of bulk up on codco, get something really, really big, but they got to go further for that. And so then we can apply this type of logic then to try and map out key areas around the colony. So this is for 4 different benthic prey, and so this is applying this idea, kind of reversing this idea that they're having to go further for higher quality prey, and use that to map out what the density of these prey must be in order to sustain for them to go that distance. And you can see that that's mapped out here in kind of a suitability. And I just wanted to point out that there's an area kind of about 15 kilometers from the colony there. There's this kind of dark area that seems to be a key area for all four species. That's an area of a lot of kelp beds, and so might be a really important area to protect. Okay, so that brings me to my second case study, which is working with the black-legged kittiwake. So this is in the Gulf of Alaska. This is working on Middleton Island, and I'm sure you're all familiar with this blob, right? This what was at the time the largest marine heat wave on record. Happened in 2014, 2015, 2016, and Fred Trombley was out there studying these birds. And again, the system, we know already that it does better during cold conditions. So when it's cold, they are able to feed on their preferred prey item, which is capelin, and the birds do much better. They have higher reproductive success. So you can see that here experimentally. So here we have the productivity, so the chicks per nest, and those are the cold years on the right-hand side, and the warm years on the left-hand side, including the years of this study here in red there. And so the productivity is much lower. And we supplementarily fed some individuals to show that this really is due to food supply. We're able to reduce or eliminate the impact of the warming event by feeding them. So we're supplementally feeding them with capelin, and then they do much better. So you can see that effect size is much larger in those warm years. And when they're in those really poor years, they're not spending much time gliding. They're working really hard. They're flapping much more, and the daily energy expenditure is relatively high. Whereas in those cold years, they're spending more time gliding and have a lower daily energy expenditure. Right. This is a little infographic that it tells you what's happening in terms of the. It seems to have gone away on me, but anyway, so in the pre-heat wave period, so there's Middleton Island at the top, so the little island out there. Pre-heat wave, the birds were doing really well. Food is abundant, so the kittiwakes forage in a small area close to the colony. You can see like it's a really small area that they just forage right around the colony. As soon as that heat wave hit, the birds' foraging range had to increase dramatically. They had to work much harder to find food and spent more time foraging over this really large area. But this persisted even 2 years after the heat wave. So in 2017, 2018, the kittiwakes were continuing to forage over this really large area despite the water temperature returning to normal. And so there seems to be this kind of persistent impact of the heat wave. And we actually get very similar results when we look at the kittiwake system. And so this is almost identical to what we were able to show for the MERS. with this path between from the PDO to diet to net energy intake to fitness. So this bottom-up impact without the limited direct impact. But in this case, there's a 2-year there's kind of a lag built into the system, so it's the PDO over 2 years rather than the PDO itself. And so there seems to be kind of this 2-year lag in the system that we didn't see with the MERS. And then just that we can do exactly the same thing that we're doing with these species and extend it to other species. This example with the tufted puffins here, and so these are birds tagged with GPS. We look at what prey they brought back, and you see this very similar results here with the gadded, so the larger prey being caught further away than the smaller prey. And so you can try and map out exactly where they're getting their prey, and then kind of apply this idea of that they're this kind of energetic trade-off to try and figure out where there actually might be hotspots versus where they're just expending less energy. Okay, and then you can do this not just at the scale of a colony, so just like that was relatively small scales, 10, 20, 30, 40 kilometers that we were dealing with before, but you can actually extend these ideas out to much larger scales, as my PhD student Danjean is doing. So these are black-legged kittiwakes from across the entire North Atlantic, and you can see there that there are energy hotspot. There's kind of energy hotspots, especially right near the range center, which we thought was really interesting. So they seem to be expending a lot of energy when they're right near the range center, and then the green kind of spread out around on average. And so maybe this tells us something about why most of the birds are going towards that range center, that there's a lot of energy there, a lot of energy available, and so they're able to expend more energy. Okay, well, I hope I've been able to convince you today that net energy intake improves our ability to use seabirds as indicators, and that energetics is a really helpful way of thinking about animals in general, not just seabirds, but this converting the biologging information they're able to get into energetics is really helpful and really useful at trying to understand some of these trends. Energy provides a key link in bottom-up effects of climate change, but in some cases there's a lag, such as responding to that marine heat wave. It seems to take several years before the birds are able to return to normal. These energyscapes also highlight hotspots for the seabirds. So I think they can be really important to try and identify hotspots and cold spots for the birds. And just to kind of propose this idea that energetics could be really helpful for conservation broadly. They can help us understand which biologgers to use, and so help us with ethics and improving our methods. They can let us measure the costs of different behaviors, such as flying versus diving. They allow us to measure the costs of climate change. I hope I convinced you to be able to link costs to fitness, to populations, to be able to find hot spots and cold spots to create marine reserves, and to be able to measure health, including, for instance, impacts of contaminants. And then just to kind of encourage you to, this is a good framework to think about these two different ways of thinking about how we sample wildlife. The classic would be this Eulerian framework, where the so kind of shifting your perspective from this classic Eulerian perspective, where your frame of reference is what you define, to this kind of biologging approach or kind of energetics approach, where you're following individuals, and that becomes your frame of reference. That I just wanted to thank the many people that have helped me. many funding sources. And lastly, I couldn't come here without showing at least one picture of a cormorant. Thank you so much for the invitation to have me. It's been a real pleasure. There's lots of really interesting things happening here. It's been really nice to talk to everyone today. And does anyone have any questions?
Thank you so very much. So we'll switch to questions and we'll start with any questions online. Roseanne, anybody? Okay, so for folks online, if you have questions for Kyle, go ahead and put them into the chat. How about questions in the room? Oh, we're going to be shy today. All right, I'll start us off. On that infographic that you showed, have you been able to see how long it took to recover the sampling ranges back down to a normal size or a historical size?
It took until 2022. I mean, for them to get back to what we were seeing in 2012 and 2013, and so it took, I guess, 9 years before they were back to normal. So the worst. So like by 2017, 2018, they still really hadn't recovered at all. 2019 and 2020 were a little bit better, as well as 2021. Then 2022, they were kind of really back to what we would expect for the ocean temperature. So these marine heat waves really do seem to persist for a really long time.
All right, other questions in the room? All right, just a second.
Kyle, I noticed that there weren't any data for 2014, the first year of the marine heat wave. Was that because no data were collected, or because it was sort of like this transition year?
Yeah. You're right. I mean, I think this is always the problem with these long-term studies that I remember, the first long-term study I ever went to work on was on song sparrows on Mandardi Island. And, Jamie Smith went out and for 7 years, nothing really happened. I mean, there was like, the reproductive success was always about the same. Survival was always about the same. So I said, well, I'm going to stop that. I don't need to do anything more. So he just went out and got a little bit of data in that 8th year. And the whole population had crashed that year. Really, oh my goodness, this was the year that I really need to be out here. I think the same thing happened to us with 2014. And so there was no funding for the station in 2014. There was the funding from USGS had dried up, and so there was no data collected in 2014. And of course, once the heat wave hit, we thought we got to really get out there in 2015, and so we were able to get some data in 2015, but the sample size was a bit smaller. But then over time, we've kind of been able to recover. But it was and same with 2020, the COVID year. I mean, our sample size is 2 birds, and so that's really small for 2020. But it's always a challenge with these long-term studies getting funding for another year. That's why there's no data from 2014.
Just as a quick follow-up, the pattern that you saw with kittiwakes on Middleton in terms of recovery from the heat wave was very similar to what we saw for marbled murrelets way down here. And I wonder if you got any insight into why the recovery was so lagged behind the actual heat wave itself.
I mean, I don't really know the answer, but I assume it has something to do with prey, and that it takes that long for, I mean, that it just takes several. So if all of the capelin are gone from the system, and it is really a capelin-driven system, like for the kittiwakes there, they need the capelin. The capelin need cold water. So it takes several years for the capelin to be able to bring up their biomass back up to something that's able to sustain the birds. And so in the meantime, they're eating whatever other junk they can find, but it's not good enough to be able to raise offspring. That's my hunch, but we don't have the forage fish data to be able to back that up.
All right, we got a question online. Go ahead, Roseanne.
Which company is the manufacturer of the tags with accelerometers that you use?
Yeah, we use TechnoSmart, I think exclusively. Yeah, TechnoSmart. So they're a group out of Italy that have really kind of, they have great tags. Yeah. There's also a group in Swansea that those daily diaries that a lot of people are using. I think those are the two main tags that people are using at the moment, but we're using TechnoSmart.
Okay, we got other questions in the room. One second.
I was a little bit slow to take in all the information on the path analysis diagrams you showed, but the numerical values that were next to the arrows, are those like similar to a slope coefficient in a like multiple regression model? And I saw like negative 0.91 for that value between energy intake and fitness. Does that mean that energy intake has a negative effect on fitness of those birds?
No, it's the other way around, and it was just the way that was set up. And so I'm trying to remember now, but it's not so it's not just I put in several. Each of those is from a principal component analysis. So rather than taking a single value, we put in several different values in there, and so this is what we think they represent. And so rather than taking ice conditions, like one measure of ice condition, because of course it's changing throughout the season. We took several different measures and then took the first principal component of that, which worked better than any given measure. And same with the fitness. So rather than having just one measure of fitness, we put in several measures of fitness. And for diet, it was the same thing. Rather than being just one prey item, it is all of the prey items together. But then the first principal component, the same is true of the energetics. And so you're right that they are they're the slope, right? But it's from that principal component analysis, and so that's why the coefficient is reversed. But definitely, high net energy intake is good, and so has a higher fitness.
All right, we've already run out of time. So just like your visit here, this is a whirlwind. We're going to end this now. And before we wrap up, Cinnamon's going to ask for another awkward photo. So my grad students that are here, before we leave, can we get one more photo and have everybody come up front and take a picture? But before we do that, everybody say thank you very much. All right, and just a reminder, we've got a social event coming up after this event. So if you have more questions over beer, you can do that as well. Thanks everyone for being here. Appreciate you.
Okay. Thank you. No. Great.
Yeah.
Yeah. Yeah.
My way I'm socially.
Am I in this photo as well? Yeah. Okay. Great. Okay. Oh, yeah.